Hi everyone, today we'll be talking or we'll be getting to know about the chapter Fundamental Unit of Life from Class 9 CBSE Biology. The first chapter that we'll be talking about here today is the Fundamental Unit of Life. What is the fundamental unit of life? The fundamental unit of life is the cell. It is the basis of life. It is the basic unit of life. This thing or this microscopic organism is able to maintain the life on its own. It can survive on its own. It can remain independent and it can continue the process of life. So they are known as the fundamental unit of life. To start with, what is cell? When we tell that fundamental unit of life is cell. So cell is the smallest living being or smallest living unit which is able to live independently, and it is also able to perform all the activities for the maintenance of life. And what are its characteristics? The characteristics are they are either unicellular, mostly they are unicellular, they can be even multicellular also, and they are the smallest living unit. They are able to carry on the life processes independently, and they are the structural and functional unit of life, and they have a variety of shapes and structures depending upon their position and also about the function that they need to perform. So the first thing when we divide or when we think of cell is that after having or getting to know about cell, the first thing comes that cell can be divided into two parts or into two types, which are two most important types. That is, first is the prokaryotes or the prokaryotic cell, and the eukaryotes or the eukaryotic cell. Now the prokaryotes, as the name suggests, they are the primitive type of cells, whereas the eukaryotes, as the name suggests, they are the more advanced and they are the recent type of the cell. So prokaryotic cells are generally or mostly unicellular. They are the most primitive type of the cells. They have a range or the diameter of ranging from 0.1 to 0.5 μm, and they reproduce asexually and through the mode of binary fission. The nucleus as a whole is absent. Instead, they have a structure known as the nucleoid, which consists of the genetic material, and they are all without a membrane. They are not membrane bound. And the genetic material which is there in the prokaryotes can be an RNA or that of a DNA. Whereas in case of the eukaryotes, they are all multicellular. Their average cell diameter ranges from 10 to 100 micrometer in diameter. They are very much evolved. The organelles are all membrane bound, including that of the nucleus. The nucleus has proper genetic material, and they contain the genetic material in the form of DNA, and they reproduce sexually and even asexually. So this is the basic differences or the basic characteristics of the eukaryotes and the prokaryotes, which we need to remember, which is very important if they ask what is a eukaryotic cell or what are the characteristics of the eukaryotic cell. So these has to be mentioned. Along with, we have to mention, depending on the marks, the organelles that is present in the eukaryotes, what functions they can perform, which is not able to function by the prokaryotes, and little bit depending upon the marks that is provided for the answer. Now first let us differentiate or let us understand the differences between the eukaryotes and the prokaryotes, because if we know the differences, we can also write their characteristics depending on the marks. Basically, when we know all the characters of each of the cell, we can do it very easily. So the first difference based on the characters, that is, first is unicellular. They are multicellular in nature. The nucleus is totally absent. Instead, they have a structure known as nucleoid, which is freely suspending, and they do not and are not membrane bound. Here they have a proper nucleus which is membrane bound and consists of the genetic material. Their locomotion takes place with the help of flagella for the prokaryotes, where here the locomotion flagella is totally absent. Lysosomes and peroxisomes are present. Here lysosomes, peroxisomes, mitochondria. Lysosomes and peroxisomes are absent. Here not even lysosomes, peroxisomes, your mitochondria, your ribosomes, your endoplasmic reticulum, everything is present, which is totally absent in the prokaryotes. And mostly they are devoid of all these organelles. Because of that, these all differences can come to. We can all talk about all these differences. See, mitochondria is absent, Golgi apparatus, chloroplast, sorry, I missed out chloroplast. Then submicroscopic size flagella, I told you. The cell wall is chemically complex. Your cell wall is quite simple. It is not that complex. Instead, they do not have a cell membrane. Here they have a cell membrane. And the permeability of the nuclear membrane is present. The nuclear membrane which is present is totally permeable. It allows inside and outside for the mobility of the materials. Here the permeability of the nuclear membrane is selected. It does not allow anything and everything to go inside and come outside. So it is a selective. Here it is totally free. Here everything can pass in and go out and come in. They have a pili and the membrane. Here they are absent. Here the transcription, the process of transcription takes place in the cytoplasm. Here the process of transcription takes place in the nucleus, because there it is the genetic material is present in the nucleus. Now let's understand more about from the eukaryotic. Under the eukaryotic cells, again we can divide it into two types, that is the animal cell and the plant cell, because there are some basic differences in animal cells and plant cells. The organelle wise, mostly they have all the organelles, but a organelle like chloroplast is present only in the plant cell, which is not there in the animal cells. Then vacuoles are more distinct and more large in the plant cell, which is mostly there. Even if it is there, it is absent. Even if it is there, it is very small and it does not have much of a function in an animal cell. So let's talk about the animal cell first. So when we talk about the animal cell, this is a plasma membrane or the cell membrane that encapsulates or that gives a shape and protection to that of the cell. So the cell membrane, which we have already told, it does not permit anything and everything. It permits only some selected things. So it is selectively permeable. Now when we talk of selectively permeable, what does it actually mean? Selectively permeable is used for membrane for the process or during the process of the method of osmosis, during the process of osmosis. Now what is osmosis and what happens when you put this kind of a selectively permeable membrane bound cell in different solutions? What are its reactions? We'll talk about it later. Okay, so this is a cell wall or it is the cell membrane that is present, or the plasma membrane, which is selectively permeable. Coming to the inside, this is the nucleus. This is the nucleus of the cell, having a core known as the nucleolus. This nucleolus is where the ribosomes are formed. This is where the ribosomes are formed, and here this is the chromatin network or the chromosomes. This contains the genetic material, and the nucleus is covered or is again encapsulated by a membrane. I told you everything is membrane bound. It is known as the nuclear membrane. This nuclear membrane has some small pores on it which allows the entry and exits of the materials. Now starting with the organelles, this is the endoplasmic reticulum, which is due to the presence of the ribosomes. It is called the rough endoplasmic reticulum. It helps in the secretion of proteins, and this, after this endoplasmic reticulum, this is the mitochondria, or this is also known as the ATP or the powerhouse of the cell, because it is here that the energy is produced. ATP is released for the functioning of the cell. And this is the Golgi apparatus, which helps in the modification of protein. These are the lysosomes, which are also known as the suicidal bags of the cell. This actually bursts and gives out when there is if the cell faces a foreign attack, or cell feels there is a foreign attack, there is a foreign body to which it has to take care of. There are also presence of the centrosome or the centromere and the centrosome. The centrosome or they are the organizing center of the cell. These are the microfilaments and these are the microtubules, and this is the centrosome and this is the microtubules. So this is more or less about the animal cell that which we will talk, or this is the detail about the animal cell, but about the cell membrane we'll again talk. Before talking or giving an idea or explanation about the plant cell, first let us know about the cell membrane and its selectively permeability. So what we are told that because of its selective permeability, there is a process which takes place. Always there is a process that takes place due to this presence of the membrane. It is the known or process of osmosis. So what happens during the process of osmosis because of this membrane? So supposedly this is a solution having solutes and solvents. Now here we have put, this is a concentrated sugar solution. Concentrated sugar solution means the concentration of the solute is high than that of this part, than this part. This is a dilute sugar solution. So the concentration of the sugar molecules or the solute molecules is more, which means the concentration of the solvent molecules is less. And here the concentration of the solvent molecules is more. Now it is a selectively permeable membrane that we have put. Now during the process of osmosis, what happens? I told you this is highly concentrated, having a less solvent particles. So the water from the higher concentration flows from that to the lower concentration. So here this part, it was having a dilute concentration of sugar, that is the sugar molecules were less, whereas the water molecules are more. So water molecules from this side flows to this side in order to equilibrate, in order to bring in an equilibrium to that of the solvent molecules. So what happens? Water from here flows into this portion having the concentrated sugar solution. In order to that, the sugar solution becomes similar throughout the tube or throughout this beaker. Now when we have talked that we have told a selectively permeable membrane, because of the selectively permeable membrane, this cell, if you put in different solution, you're going to see differences in the cell happening. If you put it in a solution having equal concentration of the solvent molecules both inside the cell as well as inside the beaker, the cell remains in more or less a equilibrium condition, that the cell remains same, even the water also remains similar condition, not the water, the solution. Now if you put it in a hypotonic solution, in a hypotonic solution you can understand that the cell, here it is shown that the cell increases, or the cell increases because due to the flow of the solvent molecules. Now why does the flow of the solvent molecules takes place? Because the concentration of the solvent molecules within the cell is lower than what is there in that of the beaker. So immediately water from the beaker flows into the cell, making the cell to blow up, or it becomes fat in structure, it becomes a little bit flaccid in nature. But when you place in a hypertonic solution, hypertonic means the concentration of the solute is more in here. That is, the concentration of the solute in the beaker is more. So obviously concentration of the solute in the solvent is less. So what happens? Water from the cells moves out into the beaker in order to make it equilibrium. So the cell shrinks. The cell gets shrunk up. So this is what happens in different types of solution. So this is very important. You must understand and you must remember what happens when a cell is put into a hypertonic solution, hypotonic solution, as well as in an isotonic solution, and what is the process of osmosis, what effects does it bring about in the process of osmosis. Now let's go on to the plant cell. In the plant cell, it is more or less similar to that of the animal cell. Here you can see a very large vacuole. Here you can see a prominent and large vacuole because it is filled with a cell sap that maintains the pressure, cell pressure against the cell wall. Now there is a presence of a rigid cell wall which was absent in animal cell. Now this rigid cell wall gives a shape as well as it protects the cell. which is absent in that of the animal cell. And here again, this structure is also present in animal cell, which was absent in present in plant cell, which was absent in animal cell. These are the chloroplasts. The chloroplast again can be divided into three types, that is chromoplast, chloroplasts and leucoplasts. Based on their functions, they have been named so. Chloroplast, as we understand the name, it helps in the process of photosynthesis because this provides with the chlorophyll for the process of photosynthesis. Chromoplasts give various colors to the fruits, flowers, etc. And leucoplasts, they are basically colorless. So they are mostly found in the areas which helps in the storage of sugar or storage of carbohydrates. So we can understand there are three types of plastids. Whereas chloroplast helps in the process of photosynthesis and chromoplast gives colors to the fruits and flowers, and leucoplasts are colorless in nature. This chloroplast, chromoplast and leucoplast, they are also found in plant cell, which are absent in that of animal cell. These are the plastids, so these are more or less similar as that of the animal cell. Now these are the type of plastids. Just now which I was talking of, it contains chlorophyll, green pigment, it absorbs sunlight during the process of photosynthesis, it produces and stores glucose. Chromoplast, it contains carotenoids and it produces the red, yellow and different pigments. They are found in flowers and fruit. And leucoplasts contain no pigment and they are used to store starch. Now let us come to discuss the importance or the differences between the cell wall and cell membrane. This is very important from the exam point of view and even for your MCQs also, because they can give one mark answer or 1 mark question MCQs based on the differences or based on the characteristics of the cell wall and cell membrane. It is present, cell wall I told you, it is present for the plants, fungi, bacteria, algae. And cell membrane is all found in the animal cells and the human cells, made up of lipid, protein, carbohydrates. They are made of peptidoglycan in bacteria, for chitin in bacteria, and cellulose in that of the plants. It is for chitin, it should be in fungi. It has a thick and rigid structure with a fixed shape. It has a thin and delicate structure and it is flexible. The shape is flexible. It is not that it is rigid and fixed like that of a plant cell or the cell wall. It protects the cell from external. It also protects and maintains the internal environment. Both have the similar function of protection, but it is rigid. This is not as rigid as that of the cell wall. It is metabolically inactive, but it is metabolically active. The cell wall is fully permeable to smaller molecules. It is selectively permeable to molecules, and functions include protection from the external environment. Functions include permeability, mobility, conduction, cell division, and they have various other functions, that is sexual reproduction, etc. If you want, you can take a screenshot of these differences between the cell wall and cell membrane, because these are very important parts of this chapter, Fundamentals Units of Life. So next we'll move on to the differences. Means if you see a plant cell and animal cell in a comparison, you can understand that mostly all of these are there for both of the cells. Only what is different is the chloroplast, cell wall and vacuole. Chloroplast, cell wall and vacuole are there in the plant cell, but it is absent in that of the animal cell. And in the animal cell you have a centriole, which is absent in that of the plant cell. Even the vacuole, which is there, it is sometimes there in the plant cell, but it does not have much of its function, or it is mostly insignificant in animal cells. So vacuole, whereas in plant cell it is very much important and very much of necessity. So this figure gives us the idea about the differences between the animal cell and the plant cell based on their organelles. And one important thing is the presence of cell wall. They don't have a cell wall. Here they will have a cell membrane. So this is in one go. We have discussed the full chapter of fundamental unit of life. We have discussed about the cell, the types of cells, the differences in prokaryotes, eukaryotes. We have discussed the differences in the cell wall and the cell membrane, the characteristics of the cell, the characteristics of the different types of prokaryotic and eukaryotic cell, and also we have understood the different types of solution, which is very important, as well as the process of osmosis. So this is it for the fundamental unit of life. In my next video will come with some of the MCQs for fundamental units of life or the cell chapter. Till then, thank you, and if you have liked my video, please like, share and subscribe to my channel. Thank you.